Types of mapping populations. There are basically two types of mapping populations, primary mapping populations and secondary mapping populations. Primary mapping populations are created by hybridization between two homozygous lines. On the other hand, secondary mapping populations are developed by crossing two lines or individuals selected from a mapping population. The primary mapping populations are of the following different types: F2, F2 derived F3 or F2:3, backcross inbred lines, doubled haploids, recombinant inbred lines, near isogenic lines, chromosomal segment substitution lines, immortalized F2, advanced intercross lines, recurrent selection backcross populations and interconnected populations. The characteristic features, merits and demerits of each of these populations are briefly presented below. The first one is F2 population. F2 population is developed by selfing or intermating for cross-pollinated species among F1 individuals in segregating populations generated by crossing the selected parents. These F1 individuals are developed by crossing two parents that show significant polymorphism for whichever type of loci you are going to score. F2 individuals are products of single meiotic cycle and the ratio expected for dominant marker is 3:1 and for codominant marker is 1:2:1. For F2 population mapping, a fertile progeny is required from a crossing of genetically distant parents. The progeny of such a cross is called the F1 generation. If the parental lines are true homozygotes, all individuals of the F1 generation will have the same genotype and have a similar phenotype. This is the content of Mendel's law of uniformity. An individual F1 plant is then self-pollinated to produce an F2 population that segregates for the traits different between the parents. F2 populations are the outcome of meiosis during which the genetic material is recombined. The expected segregation ratio for each codominant marker is 1:2:1. That means homozygous like P1 is to heterozygous is to homozygous like P2. It is a disadvantage that F2 populations cannot be easily preserved because F2 plants are frequently not immortal and F3 plants that result from their self-cross are genetically not identical. The merits of F2 populations includes the best population for preliminary mapping, requires less time for development and can be developed with minimum efforts when compared to other populations. The demerits includes the linkage established using F2 population is based on one cycle of meiosis. F2 populations are of limited use for fine mapping. Quantitative traits cannot be precisely mapped using F2 population as each individual is genetically different and cannot be evaluated in replicated trials over locations and years, and it is not a long-term population, impossible to construct exact replica or increase seed amount. Next is F2 derived F3 population. F2:3 population is obtained by selfing the F2 individuals for a single generation. This population is suitable for specific situations like mapping, quantitative traits and recessive genes. The F2:3 family can be used for reconstituting the genotype of respective F2 plants if needed by pooling the DNA from plants in the family. The demerit of F2:3 population is like the F2 population, it is not immortal. The third one is backcross or BC. Backcross populations are developed by crossing the F1 with one of the two parents used in the initial cross. To analyze specific DNA fragments derived from parent A in the background of parent B, a hybrid F1 plant is backcrossed to parent B. In this situation, parent A is the donor of DNA fragments and parent B is the recipient. The latter is called the recurrent parent. Two goals are achieved during this process. Unlinked donor fragments are separated by segregation and the linked donor fragments are minimized due to recombination with the recurrent parent. To reduce the number and size of donor fragments, backcrossing is repeated and as a result, so-called advanced backcross lines are generated. With each round of backcrossing, the proportion of the donor genome is reduced by 50%. Molecular markers help to monitor this process and to speed it up. Lines that are identical, with the exception of a single fragment comprising one to a few loci, are called nearly isogenic lines or NIL. The generation of NIL involves several generations of backcrossing assisted by marker selection. To fix the donor segments and to visualize traits that are caused by recessive genes, 2 additional rounds of cell fertilization are required at the end of backcrossing process. If 2 NILs differ in phenotypic performance, this is seen as the effect of the alleles carried by the introgress DNA fragment. Like an F2 population, the backcross populations require less time to be developed but are not immortal. However, the recombination information in case of backcrosses is based on only one parent, F1. The specific advantage of backcross population is that the populations can be further utilized for marker-assisted backcross breeding. Backcross breeding is an important strategy if a cultivar has to be introduced a single trait, for example, resistance. The next one is backcross inbred lines or BIL. Backcross inbred lines are developed by backcrossing the F1 from a cross between 2 homozygous lines to one of the parents and continued selfing with the BC1 F1 progeny to obtain homozygous lines. For example, a set of 98 cross inbred lines in rice by backcrossing the F1 from the cross Nippon bear japonica and Casalanth indica to Nippon bear and continued selfing of the BC1 F1 progeny to obtain BC1 F5 lines. A possible advantage of BLI may be the increased frequency of the alleles contributed by the parent used for backcrossing. Next is doubled haploids or DH. Chromosome doubling of a pollen culture derived haploid plants from F1 generate DHs. The suitability of double haploid progenies for mapping project has been demonstrated by Lefebvre and his co-workers in 1995 in pepper. DHs are also products of one meiotic cycle and hence comparable to F2 in terms of recombination information. The expected ratio for the marker is 1:1 irrespective of genetic nature of marker, whether dominant or codominant. The merit include DHs are permanent mapping population and hence can be replicated and evaluated over locations and years and maintained without any genotypic change, then useful for mapping both qualitative and quantitative characters and instant production of homozygous lines, thus saving time. The demerits include the recombination from the male side alone is accounted. So, it involves in vitro techniques. Relatively more technical skills are required in comparison with the development of other mapping populations. Often suitable culturing methods or haploid production methods are not available for number of crops and different crops differ significantly for their tissue culture response. Further, pollen culture induced variability should be taken care of. The next is recombinant inbred lines or RIL. Recombinant inbred lines are developed by single seed selections from individual plants of an F2 population. Because of this procedure, these lines are also called F2 derived lines. Single seed descendant is repeated for several generations. At this point, all of the seed from an individual plant is bulked. For example, a F3:4 R1 population underwent single seed descent through the F3 generation and was bulked to develop the F4. This population of seed can be grown to obtain a large quantity of seed of each individual line. Importantly, each of the lines are fixed for many recombination events. The following table shows the R1 population level of inbreeding and percentage within line homozygosity at each locus. In case of F3:4, the percentage is 75%, F4:5, it is 87.5%, F5:6, it is 92.25%, F6:7, it is 96.875%, F7:8, it is 98.4375%, and finally F8:9, it is 99.21%. These lines have several uses. First, they can be used to derive a map because it essentially is an eternal F2 population with unlimited mapping possibilities. Additionally, these lines can be scored for morphological traits such as disease resistance or flower color, or quantitative traits such as yield or maturity. This morphological trait data can be compiled and those traits can be placed on the developing molecular map. These lines are especially powerful for analyzing quantitative traits because replicated trials can be analyzed using identical genetic material. The quantitative trait data can be used to determine if any molecular markers are closely associated with those traits. RILs also equalize marker types like DHS. The genetic segregation ratio for both dominant and codominant marker would be 1:1. RILs developed through brother-sister mating requires more time than those developed through selfing. The number of inbred lines required is twice in case they are developed through brother-sister mating compared to selfing, particularly when linkage is not very tight. The advantages of RILs are once homozygosity is achieved, RILs can be propagated indefinitely without further segregation. Since RILs are immature population, they can be replicated over locations and years and therefore are immense value in mapping QTLs. RILs obtained after several cycles of meiosis are very useful in identifying tightly linked markers. RIL populations obtained by selfing have twice the amount of observed recombination between very closely linked markers as compared to population derived from a single cycle of meiosis. The demerits are it requires many seasons or generations to develop and is relatively difficult in crops with high inbreeding depression. The next is near isogenic lines or NIL. Near isogenic lines are generated either by repeated selfing or backcrossing the F1 plants to the recurrent parents. NILs developed through backcrossing are similar to recurrent parent but for the gene of interest, while NILs developed through selfing are similar in pair but for the gene of interest, however, differ a lot with respect to the recurrent parent. Expected segregation ratio of the markers is 1:1 irrespective of the nature of marker. The merits include like DHS and RILs. NILs are also immortal mapping population, suitable population for tagging the trait wherever such population is available, and NILs are quite useful in functional genomics. The demerits are it require many generations for development. Directly it is useful for molecular tagging of the gene concerned but not for linkage mapping. Linkage drag is a potential problem in constructing NILs which has to be taken care of.